Okay, so our next speaker is Dr. Elinor Sullivan from the University of Oregon, and she will speak about early environmental risk factors for mental health disorders.
Good morning. Can you hear me okay?
Perfectly.
Great. First of all, thank you to the organizers for inviting me to speak this morning. I wanted to talk a little bit about how the early environment influences both brain health, but then later risk for mental health disorders. So we know a number of prenatal factors are associated with risk for mental health disorders, including known factors such as toxicant exposure and tetrodotoxins, but also maternal obesity, maternal depression, poor nutrition, and increased maternal stress have each been linked to increasing the risk of mental health disorders for the offspring. In this talk, I'm going to focus largely on maternal obesity and maternal nutrition. And we know from a series of studies that maternal obesity has long been shown to be associated with increased risk for childhood and adult obesity and metabolic disease. But more recent evidence indicates that maternal obesity is also associated with risk for children developing anxiety, autism spectrum disorder, attention deficit hyperactivity disorder, emotional difficulties, cognitive problems, and eating disorders. So we wanted to try to examine this in a non-human primate model where we're able to disentangle some of the comorbidity of these early environmental exposures and focus here solely on mom's obesity and diet and look at how those factors affect mental health related behaviors. And to do this, we placed animals, they either remained on their control diet or they were placed on a high-fat Western-style diet that was made to mimic the average American diet. This diet is high in saturated fat and high in sugar. As with humans that consume the average American diet, the animals tended to have an increase in adiposity in response to the diet, and they had an increase in their insulin area under the curve, suggesting they were less sensitive to insulin and had some slight impairments in glucose regulation. Also, to further clarify, these were adult female Japanese macaques, and they were placed on the diet for about 2 years prior to pregnancy. So this is kind of a chronic high-fat diet, Western-style diet exposure, just as we were trying to mimic what happens in humans. So again, these are the two diets, and the adult females continued doing this diet throughout conception, during gestation, and through the lactation period. As you can see in our control group, we get a nice variation in percent body fat, with the majority of animals having a lean percent body fat of 10 to 15%, but some animals drifting up into 30% or 35% fat. When we placed animals on the Western-style diet, now our histogram shifted, so we have some animals that remain at a healthy 10 to 15% body fat, but we have many more that are showing up at 30, 35% fat, and some moving into 40 to 45% fat for their adiposity. So this allowed us to look at the animals and characterize the offspring based on mom's diet, but also her metabolic state, and try to look at these two variables separately. The offspring were weaned, and they either stayed on their mother's diet, becoming control offspring, or Western-style diet offspring, or at weaning, we switched the diet of the offspring so we could see what would happen if the mother ate a healthy diet, but the infant was fed an unhealthy diet at weaning. And so those would be our control Western-style diet offspring. And perhaps the more important group is what happens if you take animals whose mothers are consuming an unhealthy diet, but place them on a healthy diet early on in childhood. And those are our Western-style diet control offspring. Our initial findings from this model, when we looked at anxiety-related behaviors in 4-month-old infants, we found that though both male and female controls would touch these novel objects pretty rapidly within about 5 seconds, our male Western style diet animals also interacted with these novel objects pretty rapidly. Our Western style diet female offspring had this increased latency to interact with novel objects. Here I'm showing you first Mr. Potato Head, who is a novel object that was specifically selected as he is potentially fear-provoking due to his ability to make direct eye contact with these infant macaques. We then exposed them to a snake, a rubber snake, and you can see again the female offspring are having increased latency to touch the snake, and then also a colorful hanging toy where now more offspring are touching it more quickly, but you can still see there's this population of female Western style diet animals that have increased latency. And interestingly, it's the same 3 animals that have increased latency across all 3 novel objects. We went on to characterize their behavior in more detail. So this is during a human intruder or novel object test, which is a standard way of assessing temperament relating to anxiety and depression in non-human primates. And we coded the number of occurrences and the percent of test time that these animals were engaged in anxiety behaviors. And as you can see, if the mothers consumed a Western-style diet, we saw an increase in both the number of occurrences and the percent of test time that the animals engaged in anxiety. And interestingly, when we placed them on the control diet at weaning, that did not ameliorate the changes. In fact, if anything, it potentially slightly intensified the differences in anxiety behavior in these animals. Some other, if we kind of dissect out the anxiety total into specific behaviors, we saw an increase in stress-related vocalizations in animals exposed to Western-style diet prenatally, an increase in active forms of anxiety, such as trying to escape from the cage, both in the number of occurrences and the percent of test time. And interestingly, we saw that offspring who consumed a Western-style diet after weaning showed increases in stereotypy. So we're seeing some effects that are driven primarily by the mother's diet, but other effects, behavioral effects, that seem to be driven by the offspring's current diet. So we were really interested in trying to explore the possible mechanisms for these behavioral differences. The main mechanisms that we're targeting here are increased inflammation, again, trying to distinguish the effects of the maternal diet versus maternal metabolic state. We went on to characterize changes in development of neurotransmitter systems that are critical in behavioral regulation. We looked at stress sensitivity and characterized the HPA axis, and also went on to look at alterations in the early postnatal environment, so maternal behavior, maternal infant behavior, and attachment. So first, our first focus was on inflammation, and in this model and in humans, we know that with chronic elevations in adiposity, we see an increase in peripheral markers of inflammation. And here in this non-human primate model, we were also able to show that there was an increase in inflammatory cytokines that the fetus was exposed to, and when we looked at the fetal hypothalamus, we saw an increase in microglial activation in the hypothalamus as well, suggesting that the mother's inflammation is affecting the intrauterine environment and increasing the inflammation that the offspring is exposed to, and that is then being transmitted to the fetal brain, increasing neuroinflammation, or at least microglial activation in the fetal brain. We believe that this inflammatory process in the brain is affecting the way that critical neurotransmitter systems are developing. Here, I'm going to speak briefly on the serotonergic system. We also have some evidence to suggest that the dopamine system is altered as well. We found that when we looked at TPH2, the rate-limiting enzyme for serotonin synthesis expression in the dorsal raphe, that, again, offspring that were exposed to Western-style diets during the prenatal and early postnatal period showed a reduction in TPH2 expression, suggesting that they're producing less serotonin. We followed this up by measuring serotonin in the cerebral spinal fluid and also found that that was reduced in offspring from Western-style diet mothers. We have gone on to characterize the serotonin's projection systems, and we do see alterations in the area 10 of the prefrontal cortex and are currently trying to examine the amygdala. So kind of to wrap up this portion of the talk, we found that offspring from Western-style diet mothers are at increased risk for developing behavioral disorders with increased anxiety in both male and female animals, but with earlier onset in the females. We saw increased repetitive behavior. This was increased both by mother's diet, but also more dramatically by their current diet consumption, and increased repetitive behaviors were largely elevated in the males. I didn't show you this data today, but we also saw increased aggression in our male offspring towards a novel peer. And when we look at their social behavior, both in a controlled environment, but also in their home cage, we see signs of social withdrawal and impairments in social behavior of both male and female animals whose mothers consumed a Western-style diet. We, again, are trying to understand the mechanisms for these behavioral changes. We do see changes in the HPA axis, such that Western-style diet offspring have elevated cortisol across developmental time points with, when we look at hair cortisol, it's about double from what the control offspring levels. We see increased exposure to inflammation, as I highlighted a few slides ago, and altered development, both of the serotonin system, but we also see changes in dopamine innervation of the prefrontal cortex as well. In collaboration with Damien Fair, we are doing both anatomical and functional MRI on these animals, and we see alterations in cortical-subcortical connectivity. And lastly, we also see changes in maternal behavior, with the Western-style diet mothers nursing their infants more and grooming them less early on. And they also tend to retrieve their infants less from dangerous situations. So some early maternal behavioral changes as well. When we further went to look at the effects of maternal diet versus metabolic state, when we look at specific behaviors, we find that actually most behaviors are being programmed by mom's diet and nutrition, but there are some behaviors that are more strongly influenced by maternal metabolic state. And we believe that both in non-human primates, but also in humans, mother's diet, metabolic state and metabolic disease, and mental health directly influence intrauterine development. We see in our non-human primate model evidence for placental dysfunction and inflammation in the placenta, less blood flow through the placenta of Western-style diet mothers. We also see alterations in maternal behavior, this direct effect of the early postnatal development. And we believe this is happening through pathways such as inflammation, potentially alterations in sex hormones, metabolic hormones, and nutrients. And these factors come together to influence the way the brain is developing. So far, we've found differences in serotonin, the serotonin system and dopamine system, also some changes in the melanocortin system that directly regulates energy balance regulation. And again, the behavioral outputs being primarily anxiety, aggression, repetitive behaviors, and impaired social behavior, which we think are behavioral indicators of increased risk for mental health and neurodevelopmental disorders in humans. So kind of our next steps were to translate these findings from non-human primates into humans. And so for the past 10 years, we've been characterizing mother's diet and obesity and looking at offspring risk for neurodevelopmental disorders. We started small with this pilot study looking at 68 mother-infant pairs and recently obtained NIH funding to recruit 300 participants. And so I will be showing you some preliminary data from our pilot study. And again, we're trying to do this in much more detail and validate this further in this ongoing larger study. Our major study goals were to characterize the changes in the in utero environment associated with maternal obesity, poor nutrition, maternal stress, and depression. We're doing lots of robust measures of maternal mental health. It's not just focusing on depression, but that does seem to be one of our strongest predictors. and determine which factors are the strongest predictors of alterations in infant and toddler behavior associated with ADHD and other neurodevelopmental disorders. At this point, we're currently characterizing the infants and toddlers up to 3 years of age. If our hypotheses are confirmed, we hope to be able to develop new ideas for the prevention and intervention to reduce neurodevelopmental disorders. So one of our first findings from these studies were that when we looked at infant affect at 6 months of age, we found that infants who had a familial risk of ADHD, meaning that either one of their parents or a sibling was diagnosed with ADHD, showed early on differences in how they would interact in these laboratory behavioral assessment tasks. So here I'm showing you data from the still face and arm restraint paradigms. And we found that infants with a familial history of ADHD showed increased negative vocalizations during arm restraint and decreased attention-seeking regulatory behaviors during still face. So we believe that this is an important clue and could be an early behavioral biomarker for risk for ADHD. I'll kind of wrap up this talk. These children are now 7 and 8 years old, and so now we've been able to actually look at this data to see if it does predict ADHD diagnosis. And in fact, this early 6-month-old measure, it strongly predicts their risk for ADHD when they're 6 and 7 years old. So we do believe that this finding that this is an early biomarker for risk for ADHD. We went on to look at inflammation during pregnancy in these females, and just like we saw in our non-human primates, it has also been reported in the literature, we saw elevations in a series of cytokines such as IL-6, TNF-alpha, and MCP-1 in our participants that were obese. And when we went to try to understand, again, look at the mechanism for these behavioral changes, so here I'm showing you again negative affect during still face, we saw that was, in addition to being related to risk or familial risk of ADHD, was also related to mom's metabolic state. So offspring from obese mothers also showed increased negative affect during still face. And when we went on to probe this relationship further, we found that this relationship between mom's pre-pregnancy BMI and 6-month infant negative affect was actually going through inflammation, and specifically third trimester IL-6. So we do believe that this inflammatory pathway that we also saw in the non-human primates is a really important driver, and again, another potential biomarker for increased risk for neurodevelopmental disorders. Interestingly, when we started we added maternal nutrition into this relationship, we saw the influence here as well. So first, we're looking at, again, negative behaviors during the still face paradigm and their relationship with pre-pregnancy body mass index. And you can see, as I reported in the last slide, that as mom's body mass index increases, we see an increase in this negative behaviors during the still face paradigm. If the mother ate 1 standard deviation below the mean of omega-3s, we saw that relationship was further exacerbated with those offspring showing higher levels of negative behaviors. So what was really exciting to us is that when we looked at moms who were eating 1 standard deviation above the mean of omega-3 fatty acids, we actually found that relationship no longer held, and offspring whose mothers had an elevated pre-pregnancy body mass but were consuming higher levels of omega-3 fatty acids were actually protected from this effect. And so we were really excited to finally have some good news to share instead of the doomsday effects of the Western-style diet and maternal obesity, that in fact there is some healthy foods that can actually ameliorate some of these changes. Just one other last slide to kind of further show that we really think that inflammation is the common pathway to ADHD and other neurodevelopmental disorders. When we looked at things like omega-3 fatty acids, we again saw that to be negatively associated with inflammation. Maternal distress and depression was positively associated with inflammation. and maternal BMI was really strongly associated with maternal inflammation, was actually our strongest factor to date, being associated with maternal inflammation. And these factors were able to predict child ADHD symptoms at 4 to 6 years of age through the pathway of maternal inflammation. So our ongoing studies, again, will be expanding the cohort to 300 women to see if we're able to validate our findings from the pilot study. We're going to further examine inflammation as a mechanism in the breakdown of self-regulation and psychopathology, and really start to dig into the mechanisms for maternal nutrition-induced behavioral programming, starting to look at epigenetics, the microbiome, hoping to add some neuroimaging measures onto this cohort. And we're also doing some cell isolation stimulation studies from the umbilical cord. Our long-term goal is to try to develop clinical biomarkers of risk for neurodevelopmental disorders and to develop early interventions, such as a dietary intervention, exercise as a potential intervention, antioxidant treatment. So far, our most promising finding to date is omega-3 fatty acid. Consumption or supplementation could be an effective intervention as well. And lastly, to help define effective prevention strategies. So at this point, I'm happy to end here. I would also just quickly like to acknowledge members of my laboratory that contributed significantly to this work, such as Jacqueline Thompson, Jennifer Bagley, and Madison DeCapo, collaborators such as Damien Fair and Dr. Joel Nigg, who are instrumental in this work, and our funding sources. And I'm happy to answer any questions.
Thank you. That was really interesting. We have a few minutes for questions. So one question I have is beyond omega-3, what do you think, or lack of such in the Western diet? Are there other features of that diet that you think are contributing to this inflammatory process? Are you trying to break that down?
Absolutely. I think that sugar specifically, but all unrefined carbohydrates is a major driver of this inflammation. We have not yet been able to break that down in the non-human primate model, but we do have really detailed nutrition measures in our humans, so we can start looking at that relationship first in the human model and then kind of bring that back to the non-human primate to be able to directly test that. I also think saturated fats, are the two biggest drivers. I don't think it's the total fat intake. I think it's the type of fat that is driving this inflammation. We know omega-3s are protective. Likely omega-6 fatty acids being in elevation are also helping to drive some of this inflammation.
And is there an interaction between the ADHD, familial ADHD vulnerability and maternal obesity? I mean, you showed them separately. I don't know if I caught that.
Right. We didn't see that in this small pilot study, but there is evidence that individuals with ADHD, both children and adults, are more at risk for obesity. And so certainly in a larger sample size, that may come out.
Oh, interesting. Okay. Any other comments, questions from the audience? Yes?
We have one from online.
Online question? Okay, we're going to try and project an online question. Could Dr. Sullivan comment on harmonization of measures or other common elements or issues addressed in related major projects, including ECHO, the adolescent brain and cognitive development, and the new healthy brain and cognitive development studies?
Yes, so I'm more familiar with the ECHO studies, and we are in close contact with many of those investigators, and we're actually really trying hard to align our efforts so that we are making common measures, especially in this ongoing larger study. We we are trying to make sure, for example, that we all send our plasma for inflammatory measures to the same core so that we can really align across centers. We're trying to align neuroimaging measures, again, so that we can expand our cohort, because really the strength here is to be able to expand the sample size and see if we see similar measures and how they validate across sites and across. One thing that's really lacking in Oregon is diversity, and so we're really interested in trying to look at how this plays out in populations with higher levels of diversity than we can do here in Portland, Oregon.
Damien?
Hello, Elinor.
Hi, Damien.
I'll just follow up on that, in that there is actually an effort right now, particularly for the brain imaging pieces of this, an effort right now in ECHO and across ABCD, and with the meeting, a kickoff meeting for the Healthy Brain Initiative next month, just before SFN, to try to coordinate the imaging kind of measurement and processing. So there is some efforts right now to do just that, at least on the imaging side, and as you know, along with kind of what Elinor has been talking about for the other physiologic measures as well.
Can you define echo? Can anybody define echo?
I don't even know.
Is it early childhood health
Outcomes?
Yeah.
Sounds good? We're buying it. So Damien, while you're there, can I ask you, I was mentioning earlier that I was a part of the biomarkers consortium, and part of the measurement question is whether there is a coordination of neuroimaging, especially across early life. Is that an ongoing effort to coordinate imaging criteria, mechanisms, scanners, whatever?
It's fascinating. It is an ongoing effort. It's very difficult, right? Because we have, as physicists and neuroscientists, everybody's extremely opinionated about the special thing. But a lot of these larger studies and larger initiatives are, by their nature, kind of these because vortex where people just start kind of following along. Like, so for example, the ABCD study sequences, I don't even know how many sites are now using them across all the different studies. Like almost all of my studies are using them. So people are starting to coordinate just because of these efforts by themselves. On the infant side, it's a lot more difficult because they haven't started yet. But the baby human connectome project has kind of filling that void a little bit, and the sequences that you're using for there are being used by lots of folks. And my guess is that the healthy HBCD will be will serve that purpose as well.
So we can return to this, but the idea of coordination and standards and measurements, how to at least get reliability as well as validity and cross comparisons, I think is something that's good to signal. Okay, thank you very much. We're ready to transition.
May I just make one statement or correction that the ECHO stands for environmental influences on child health outcomes.
Okay, that's good. Great. Thank you very much. That actually worked remarkably well. I had trepidations, I have to say, for the long distance presentation. It worked beautifully.